Okay, thank you, Dr. Muhammad, for the nice introduction. Can you hear me well, or?
Yeah, yeah, you're set.
Okay, hello everyone. It's my pleasure to give a part of my research through the third IPGG. Today I'm going to give you a talk on identification and validation of high LET hotspot genomic regions harboring stem rust resistant genes in wheat. Um, this is my agenda. I will start with its introduction and then genetic variation in stem rust resistance. After that, I'm going to give you some result on genome-wide association study and genetic validation of genomic regions harboring SR38. Finally, and I will finish my presentation with our protocol for selection of the most promising stem rust resistant genes with a small conclusion. Actually, this part of work I did it when I was postdoc at Prof. Dr. Stephen Banziger lab, but has recently published in Frontiers in Genetics. So to start with the introduction, stem rust is a very serious disease caused by Vaccina gramineae, which causes a significant reduction in wheat production. This disease can be controlled by two different ways: chemical spread or genetic distance. Spread, as you know, it's harmful on the long term, and in addition to its high cost. So an alternative solution that we could use the genetic resistance to identify most promising wheat cultivar in a combination with high yielding. Um, as a fungal disease, stem rust resistance follows gene for gene theory. The gene for gene hypothesis states that for each gene controlling resistance in the host, there is a corresponding gene controlling pathogenicity in the pathogen. So genotypes may lose their resistance on the long term, and we should always fighting this disease by screening many genetic resources of wheat to um, identify new genotypes that resist the new races. Therefore, efforts for breeding to stem rust resistance, along with identifying genes controlling the resistance, are still needed to produce stable genotypes. Um, Evaluating new genetic resources, as I said earlier, plus the advances in genetic analysis, such as genotyping by sequencing, genome-wide association study, and QTL analysis, paved the way for identifying the promising resistant genotypes to stem rust races. As this work was done in Nebraska, so we were looking for solution against QFCSC stem rust race. Because this is the common race in Nebraska, and the efforts are ongoing to fight this race. Actually, there are few studies were done to reveal the major and the minor genes against this race. We used to understand the genetic variation in stem rust resistance. We used a panel, association panel, consisted of 212 randomly selected genotypes from F3 6 lines, and this work was a part of Dr. Shams from Sadat University. And actually, this is in the right side. This is the breeding program of Dr. Stephen Panziger. So he started crosses of from 800 to 1,000 crosses. As you see, two parents, and then um, to produce F1, and so selection is ongoing in each year based on the criteria which is mentioned here: grain yield, grain volume, weight, disease resistance, and plant height and maturity until F6. So the actually the population that we used was very highly diverse because each line came from two different parents. All plant material were evaluated in their resistance to stem rust race, QFCSC, and we used two susceptibility checks and two resistant checks. We done our experiment in four replications with randomized complete block design. The evaluation was done at seedling stage, and we inoculated artificially 3 leaves per genotypes with pressurized atomizer. Um, and we use Stackman scale. It is visual score. As you see, this is the symptoms of a stem rust. And as you know, as I said, gene for gene theory. So we know the IT phenotype has genes. Actually, when we started this population, we from the pedigree information, we know that our population have uh, SR38 genes and SR24 genes. So we expected actually in our GWAS to find these 2 genes when we do the statistical and genotypic analysis. The Stackman scale was converted to 0.9 for the phenotypic analysis, and we have 3 categories here. From 0 to 3 is resistant genotypes, from 3 to 6 is moderate resistant, and from 6 to 9 susceptible genotypes. And then the same material were used for genotyping using genotype biosequencing. We extracted the DNA extraction from the whole set of genotypes and we sent them from Kansas State University, at which we done the GBS by Dr. G.C. Poland. And then my colleague, Dr. Vikas Balankar, did the SNP calling and the bioinformatic work. And finally, we got about 17 SNP that was used for genome-wide association study. Here, this is, as you see, this we have really huge variation in the stem rust resistance. This group is highly resistant, this moderate resistant, and we got some of susceptible genotypes. It was worthy to investigate the genetic diversity of the population. So actually, the first step before working on that population, we did extensive genetic diversity, and this paper was published in 2018 from our group by Dr. Shams. Was a part from Dr. Shams' PhD thesis at this time, and we found the whole population were divided into 3 possible subpopulations. And we were interested, after evaluating the stem rust resistance, to understand the variation within the 3 subpopulations. So as we see here, and this is a population structure, we have 3 colors, each color represented subpopulation. And in the right side, this is chart is the genetic variation in stem rust resistant in each subpopulation. So if you see here, there's a population in red and population in green, um, hold a very resistant genotypes, and followed by subpopulation 3. And this part is very important in our selection programs for selecting the most promising genotypes as the candidate parents. So please bearing in mind this point when I come later to the genetic analysis, because we use this information to select the promising resistant genotypes. Come to our interesting result in the genome association study. This is the GWAS table. As you see, we have as a total, we found 84 SNP markers associated with stem rust resistance, and this is the p-value. As you see, very low p-value, and this is phenotypic variation explained by marker. We have very high percentage, as you know, is more 10% R squared is considered major QTL. So here we found some big good SNPs with that has major effects on the stem rust resistant genes. And we found that the 84 SNPs were distributed in on 4 chromosomes. The highest chromosome was 2A. This hold about 60 SNPs, and we found this is our known SNP positions. So if you know that in the SNP calling of genome-wide association study, we may find some unknown position in SNPs which we don't find in the reference genome. So this is the total SNPs that we found. This is the Manhattan plot, as you see. This is a huge or big signal in the chromosome T2A. And also we have some signals in 7p chromosomes, and this is the unknown chromosome, and this is 1p chromosome. Um, it was worth investigate while that to investigate the high LED. So here, if I go back to the slide, so we have like 84 SNPs. Some located on the same chromosome. So it was very important to know the relationship among the SNPs which were located on the same chromosome. In the chromosome 1b, we found only two SNPs in high LED. This is high LED. It was very interesting that all the SNPs which were located in the 2A chromosome were in LED. On the 7 chromosome, we found like 7 SNPs in complete high LED. And also in the unknown chromosome was also in high LED. I know that we have students here, so I just give a small hint about the LED. The linkage is disequilibrium is the non-random association of alleles of different loci. So if we have two SNPs and the SNP one is A, C in variation, and the SNP two is T and G. And imagine that A and T are the resistant alleles. So if A and T, in this case, tended to appear together in the same genotype. So imagine that we have 200 genotypes. So it is like here, sorry, from A, when there is A, so there is T, as you see here. And when there is C, there is G, the susceptible alleles. And maybe we find some genotypes which don't follow the case, but this measures the LED. If the complete LED means that the SNP one is A, all genotypes A, and SNP two is T, for example. Of course, this is very important to know because it gives important information if these two SNPs are different QTLs or separate QTLs. So in this case, it is complete is one QTL. So all this SNP represented one QTL and can be co-inherited together from generation to generation. This is the same case. This is maybe uh, lower case than those. This is here. There is no LED, so this is dependent. This SNP is a different QTL. This is different QTL. These two SNPs in high LED tended to co-inherit it together in the next generation. Um, as I said, we have unknown SNPs. So we were interested to know if those unknown SNPs belong to any of the significant SNPs which were located in other chromosome or not. So actually we did LED between the unknown SNPs with the significant SNPs on chromosome 1p and significant SNPs in chromosome 7b and 2a. And actually we found that the unknown SNPs, unknown positional SNPs, uh, were in high LED with those SNPs located into A. So we suggested that these unknown SNPs were a part of this genomic regions. And we considered in our work that these unknown steps belong to a chromosome. So this is another feature of performing LED analysis that you can know the actual position of unknown SNPs in which located in any of wheat chromosome. And to understand the genetics and identifying the most resistant genes. So here we said in our in our paper that the marker that were insignificant LED will be considered as a genomic regions. So this is all significant steps as you see here in my slide. We have here one genomic region, okay, and we have two genomic regions. Genomic region 2, which has the SNPs in SNP marker in 2A chromosomes and unknown SNPs. So as a total, we have 71, and um, the significant markers in chromosome 7p. And actually, when we reviewed the genes and markers that were previously published in the stimulus resistance, we didn't find in anyone published SNPs on that chromosome 7b. Maybe one study on DART marker with Dr. Ibrahim Basyouni from the Manhoun University. He was also a member in Dr. Stephen Banziger group, but for GBS and SNPs, maybe, yeah, maybe let me see a few papers. We were one of them. Okay, so please remember this figures. So numbers GR1 we have 2, and GR2 we have 71, and GR3 we have 8, because we will use such numbers in next slides. Um, as I said earlier, that most the genomic region in chromosome 7b was new for us, so we did gene annotation in plant ensemble genes, so to know if these SNPs are a part of genes or not. So we found that these three blue SNPs were a part of three genes. Okay, and these genes were um, or encode to cytochrome P45 enzymes, and these enzymes um, were published before with their strong association with disease and fungal disease resistance. So it's like we confirm our results with gene annotation. This SNP, which in red, we found that it is located within a gene model which include two P-loop containing nuclease triphosphate hydrolase, which also one of the most important protein that can resist the fungal disease in wheat and rice and barley. So here we consider this is a new putative genomic regions in wheat which were not published before or elsewhere. Uh, Genetic validation of genomic region for harboring stem rust SR38 gene. Um, Actually, as I said before, that we expect to have two genes because these two genes were in the parents that we started the crossing of our population. So we genotyped the whole population with the two specific markers, SR38 and SR24. And what was interesting that the single marker analysis, we had very low p-value with SR38 genes. Meanwhile, we didn't find any significant associated with SR24. And when we look to the phenotypic variation explained by marker, we found that this marker explained 56, or let me 57 percentage of the phenotypic variation, which is very large, and it can explain the variation in the resistance in this population. So this is like a specific DNA marker. So we also this is also a publication in molecular marker dissection of stimulus resistant genes in Nebraska breed wheat germplasm. We published this paper in 2019, but it was in a different population. And we also found SNPs associated with SR38 located on 2A chromosome. And also we did a SSR specific marker, and we found also SR38 and some SNPs associated with stimulus resistance in that population. And here this is from this paper, Nature paper by Murad et al. 2019. Um, and this is here, this is the SSR marker. SR38 was in a high LED with some of SNPs markers that were located into A. What we did the same in the new population. We have, this is our marker, SR38 gene, and we did LED with the all SNPs located on chromosome 2A. Why? Because SR38 is a stimulus resistant gene located in 2A chromosome. So we also find high LED between this specific marker, specific gene, and the all SNPs found in the genome-wide association located in 2A chromosome. So when we compared our genomic regions with the genomic regions reported or published in our Nature scientific reports in 2019, we found 8 common SNPs between the two genomic regions. So it is a kind of a strong validation, because we used two different populations with two different genetic backgrounds, and each population came from a crossing among different parents. So we can say that we have here about I think 80 SNP marker associated with SR38 gene. And of course, this marker can be used to CASP marker, which is safe and very accurate type of DNA marker, and can be used for marker assisted selection. And I believe that Steve already converted some of those marker, and he used also in his breeding program for catching SR gene or screening SR38 in the for his populations. So come to the selection for the most promising stem rust resistant genes for upcoming breeding program. So most of previous literature studies on stem rust, they focused on phenotypic selection. This is one type that you do screening and zero score, and this is resistant genotypes without do genotypic or do any genetic analysis. Also, other part of this previous publication that interested to identify genes in associated with stem rust resistant genes without go into the genome of each selected genotypes. So here in our criteria or our selection, we actually incorporated many criteria together to identify the true and promising stem rust resistant genes. Actually, this protocol we used in our PMC genomics published in the I think this year, yes, 2021. And as you know, the effect of genotype environment interaction hinder the selection, phenotypic selection, and genomic selection. But in this study, we really overcome this problem by identifying a protocol or reporting protocols that we use genetic diversity analysis, GWAS, phenotypic selection all together to identify the most promising genotypes, high yielding genotypes that can be integrated in a breeding program for more than one location. So here we have 9 locations in these papers, no significant correlation between yield. So zero, maybe very few, no significant correlation as I remember. And by using or utilizing the features of genome-wide association mapping and diversity analysis, we come up with very few genotypes which can be used in more than location. We use the same protocol in the same rust resistant paper. So the first one, the phenotypic selection, it's very important. We select those genotypes that were scored from 0 to 1. Okay, so highly resistant genotypes, and as a result, we had 65 genotypes. So this was the first step, and we excluded all genotypes, of course. The second stage, the second criteria, we go deep into the genome of each genotype, selected genotype. So we have 65 genotypes. This is from here from 1 to 65. The below chart, the black chart is the phenotypic score. So all genotypes here is from 0 to 1. Um, the second chart is we looked to the resistant allele in each genotype. So we had GWAS study which revealed 84 resistant allele. So we looked why we don't look those steps on each selected genotypes. So here, as you see, the orange is the genomic region 1, which 2 SNPs, and the gray one is genomic region 2, which included 71 SNPs, high LED SNPs, and finally GR3, which included um, 7 SNPs located in chromosome 7b. So what we found was very interesting, that all genotypes included the genomic region number 2, 71, except genotype number 1. Okay, and we have here from genotype 44 to genotype 65, we have those genotypes had all the resistant allele that we that we found in our GWAS study. So now I would like to minimize the selections. Of course, if I go deep and I would like to select fewer resistant genotypes, so which one should I select? So here we decided to select this one, which has only 2 genomic regions and hadn't the genomic region number 2 and those genotypes. So why? So we looked into the phenotype. So this seems very resistant genotypes, but it hadn't the SR genomic region 2, which is included 7 to 1. And okay, we said so this genotype is very resistant and linked to this high genomic regions. So we decided to go the criteria number 3 to investigate the genetic distance between these genotypes and those genotypes. So as you know, genetic diversity, we measured the genetic distance between those genotypes, because you know, if you have 2 genotypes with high genetic distance, it means that it may have different genes. So if I do crossing, so I may get a hybrid with more resistant genes that existed in the 2 parents. So and here is the genetic distance. We find that these 2 genotypes very similar to each other, and both of them had 48 resistance allele. So if I did crossing between them, it will be useless. I will not gain any superior genotype for stem loss resistance. So by looking to genetic distance, we find these genotypes which hold the 2 genomic regions, um, and these genotypes has a high genetic distance with or let me to say the genetic distance between the resistant genotypes were from 50 to 61 genetic distance. So if I select this genotype, with this genotypes, this is the genetic distance was 0.61. So it is high genetic distance and low genetic similarity. So if I and this one hold or had 84 resistant allele, and this is one maybe without the GR2 genomic region. So crosses, if I did the cross between these two parents, I may get a better hybrid that have higher resistance for the stimulus gene than its parents. So by looking to these three criteria together, so we select based on the phenotypic, the target resistant allele, and the genetic distance. So by this way, we can really select the most promising and fruitful crosses that can be parents that can be crossed together to produce stimulus resistant genes, stimulus resistant cultivars. This work was published in Frontiers in Genetics last month, and was titled Identification and Validation of High LED Hotspot Genomic Regions Harboring Stimulus Resistant Genes on 1p, 2a, and 7p chromosomes in wheat. You can get the detailed informations about this in the website of journal, and this is free access, so everyone can download it. So come to the conclusion. We found three LED genomic regions controlling important SR genes associated with stimulus resistance, and in particular, the genomic region harboring SR38 was identified and validated, and this is very important for marker-assist selection and future breeding program. The validated SNPs in these regions can be converted to CASP marker, which can be used for marker selection for SR38, and I think now Steve used them for his program. Moreover, important new SNPs, especially those located in 7p chromosomes, were identified. This is very important as we find new SNPs harboring new genes. This will lead to genetic resistance for this race. The gene annotation analysis revealed putative genes associated with fungal disease resistance. Actually, all steps that we found in GWAS analysis were located within gene models that have a strong association with fungal disease resistance, and this is really gives the power of genomic association study to identify the QTL and gene associated with target traits. And finally, the interesting combination between phenotypic selection and GWAS and genetic diversity plus population structure identified the most promising wheat-resistant genotypes, about 23 genotypes, which can be used for future breeding program. And finally, I would like to express my gratitude to Dr. Stephen Banziger for supervising this work, and Shams El Tahir. And we worked together closely together in this project, and Vikas Balamkar and Dr. Ahmed Al Quda. And this is the interesting groups where I spent 2 years during my stay in USA. Was wonderful time to me and Dr. Stephen Waglu. And thank you so much for listening. And it's my pleasure to have your questions.
Okay, yes, thank you, Dr. Ahmed, for valuable presentation and for have a very important issue. Actually, it's like a paramount issue, STEAM trust. Um, thank you. Any question, please raise your hand. You can write down. So yeah, so as I say, thank you, Dr. Ahmed Sallam, for your nice presentation, and your great efforts. Thanks a lot, Dr. Azza Sami Hussein. Many people actually, Dr. Ahmed, Inas Tahir, and Muhammad Abu Zaid say yes, thank you for your nice presentation and great work. Uh, let me scroll down here to see if there is more comments or yeah. So any questions, any comments? Please go ahead. Well, yeah. At that point, thank you, Dr. Ahmed,
Thank you,
and
Dr. Muhammad.
you are, yeah, you are welcome. So I have to finally thank all the three presenters today, actually great presenters. Dr. Dr. Amir Ibrahim from Texas A&M University, United States of America. Of course, Dr. Peter Steven Benziger from University of Nebraska, Lincoln, United States of America, and of course, Dr. Ahmed Sallam from Asit University, Egypt. Thank you all for your attendance, and don't forget tomorrow is the last day, and don't forget to do like registration on time. And yeah, if you have any question, any comments, please go ahead and write down. And yeah, have a great day. Okay, bye bye.